So our second lecture in this Integrative Metabolism Learning Unit really sort of was an opportunity to review and revisit kind of type 1 and type 2 diabetes. We've talked already a little bit about metformin. We talked about it as an inhibitor of the electron transport chain, but kind of see how it comes into play here as a sort of first-line treatment for type 2 diabetes. And then also learn some new hormones. So again, we're going to learn about AMPK, leptin, Ghrelin, Adiponectin, PYY, and then talk about non-alcoholic fatty liver disease. We're actually going to mention that kind of first here as we kind of introduce this section. So again, all of those old hormones, we'll kind of review those that are important to kind of still think about here. And again, this chapter, this learning unit is all about sort of integrating some new concepts with previous knowledge. All right, so the first thing I want to talk about is just this concept of what non-alcoholic fatty liver disease is, why it's important, why it is sort of something that you're hearing a lot about. So metabolic syndrome is one of these things that tends to be coinciding with people who have this non-alcoholic fatty liver disease. Now, why we call it non-alcoholic fatty liver disease is fatty liver disease can be fairly common with alcoholics. So we're seeing a lot of the same sort of pathophysiology, but different sort of causative agent. So some stats that are a little bit staggering here, but 25% of the world's population is thought to have non-alcoholic fatty liver disease. Actually, I'm not certain if that's just non-alcoholic or if it's just fatty liver disease in general, but about 30 to 40% of the population in the United States, that is crazy how high that is. I mean, I know it's not 50%, but that's almost half of the population if we're thinking about getting towards that 40% mark. That's like 70 to 100 million people in the United States have fatty liver disease. So kind of walking through sort of the stages of fatty liver disease, right? So again, here's your liver. We've talked a lot about the importance of the liver. I hope I've convinced you of all of the important biochemical functions that your liver has to do. So it's important that your liver be able to function properly. So what begins to happen in this kind of progression of fatty liver disease is you begin to have deposits of fat in your liver, which can cause liver enlargement. Okay, so again, these are going to be triglycerides that are kind of deposited in your liver, not supposed to be there. But again, there could be a variety of different hormonal imbalances or toxicities or a lot of reasons that could be causing this deposition of triglycerides in your liver. Now, this stage is largely asymptomatic, so we don't see a lot of symptoms that we have here. But again, that can be a reason why, there's so many people in the United States that likely have this, but again, most of them are asymptomatic. The good news is if we look at sort of this progression down here, so again, we've got a healthy liver into our fatty liver, all of these blue arrows indicate things that are reversible by diet, exercise, weight loss. They even talk about bariatric surgery here. But importantly, the idea is that this process is reversible, even up to and including this inflammatory stage. So again, what kind of happens as you go through this progression is these accumulations of fat deposits ultimately cause some inflammation. That inflammation can kind of lead to a fibrotic state. And again, that can lead to even more problematics with liver cirrhosis and ultimately can cause a cancerous state. So from a diagnostic standpoint, the gold standard for diagnosing non-alcoholic fatty liver disease is a biopsy. But because so many people are literally asymptomatic and sort of live in this very reversible stage, there's risks to liver biopsy, and the risk is too high. So they tend to not do a lot of testing diagnostically for this. If we think about sort of some causes from a dietary standpoint, omega-6 fatty acids, which remember we learned about those. Those are pro-inflammatory. Those are a lot of your seed oils. These contribute heavily to non-alcoholic fatty liver disease, as does elevated levels of fructose consumption. So we've spent a lot of time in our class here sort of talking about the differences between lots of different macronutrients and then the problems that we have, particularly with omega-6 fatty acids and then fructose. So one of the things that's just important to sort of highlight here is some of the comorbidities that we see with non-alcoholic fatty liver disease. So the things that I've highlighted in blue are three things that if these three things are present in a patient, we refer to that patient as having metabolic syndrome. So those are obesity, type 2 diabetes, and then elevated blood pressure. So again, those three comorbidities will be classified as somebody having metabolic syndrome. Some other comorbidities, and remember, a comorbidity just means an underlying condition that makes it more likely for someone to have sort of another disease here. Or these diseases are things that tend to be found or grouped together. Hyperlipidemia, right? High levels of circulating lipids in the blood. We tend to see, again, insulin resistance, right, with somebody who's a type 2 diabetic. And so that is going to impair their fatty acid metabolism. And so in this case, we're going to see elevated levels of fatty acids in the bloodstream. Hypothyroidism is a comorbidity for non-alcoholic fatty liver disease, as is obstructive sleep apnea, and again, elevated transaminase activity. So we just learned about ALT in our previous discussion of the Cahill cycle. So two enzymes, these are two liver enzymes. If you ever have a liver panel done, they'll be looking for ALT and AST, are two transaminases. And so again, elevated level of this transaminase activity is a comorbidity for non-alcoholic fatty liver disease. So ALT is for alanine transaminase, and AST is for aspartate transaminase. All right, kind of summarizing what we know about type 1 and type 2 diabetes. And so I'm going to kind of go back and forth here. These numbers, things that are numbered the same, sort of are kind of related to one another. So here you, again, can see a lot of the symptoms that we see with patients who have diabetics. The ones that are in blue are going to be ones that are more common in someone with type 1 diabetes, again, but a lot of these are very common in both type 1 and type 2 diabetes. So again, type 1 diabetes tends to be found in children and young adults, whereas we used to call type 2 diabetes age onset because it tended to not appear until much later in life. But now, again, we see it in all age ranges, and we really refer to this as a lifestyle disease because it is onset much earlier in life because of lifestyle and dietary choices. All right, so both of these are fundamentally an inability to regulate blood sugar levels. And with somebody who's a type 1 diabetic, that's because they're unable to produce insulin, where with somebody who is a type 2 diabetic, they have an inadequate response to insulin. And as we'll see, as we kind of discuss this a little bit further, there is actually sort of a suppression of the ability to produce insulin in someone who's type 2 diabetic. So sort of early on, we do have sufficient insulin production, but there's an inability to listen, that concept of insulin resistance. But later on with individuals who have type 2 diabetes, and this is at the point where it becomes very difficult to reverse this, we actually have a 50% reduction in actually the number of beta cells. Remember, it's the beta cells that are in the pancreas that are responsible for producing insulin. So after an extended period of insulin resistance, you actually will have a decrease in the number of beta cells. And so you have both necessarily insulin resistance in these patients as well as decreased levels of insulin. So oftentimes it is not uncommon for people with type 2 diabetes to eventually have to take exogenous insulin because they're just unable to produce sufficient amounts. Let's contrast that with sort of the pathophysiology with what happens with a patient with type 1 diabetes. And in that case, they really have a destruction of beta cells in the pancreas. And there's thought to be both genetic and environmental contributors to this. The genetic piece basically just comes from the fact that if you have a parent with type 1 diabetes, you have a 1 in 20 chance of developing diabetes yourself. And again, it's not like it's a flip of the coin type of a chance, but there is this predisposing genetic factor. And then again, we're still sort of trying to understand a lot of the environmental contributors here, but it is a big question is whether or not gluten has played a contributing role in sort of an environmental exposure. And this is sort of paired with the fact that about 6% of type 1 diabetes patients also are celiac patients. And so there really seems to be an autoimmune sort of component to this. And truly, one of the ways that they diagnose and differentiate type 1 versus type 2 diabetes is looking for those autoantibodies. And so one of the antibodies that they do look for is anti-GAD antibodies. So again, we really haven't talked too much about sort of the biochemistry that happens with this, but this enzyme, when you start developing antibodies for it, that is sort of a marker for type 1 diabetes. So some other things to kind of highlight here. I said this in the beginning, but just to highlight here that both type 1 and type 2 diabetes really are an inability to control blood sugar level. Again, either because you cannot produce insulin or you do not properly respond to insulin. So when we think about sort of risk factors and what we see here for type 2 diabetes, obesity is an absolutely a risk factor. BMI greater than 25 puts you at an increased risk for type 2 diabetes, and decreased exercise, that's problematic. If we just sort of look at sort of the trend and remember the difference between correlation and causation, so this is just a correlational observation here. But if we look at, since the 1960s, the increase in BMI, it correlates very strongly with the increased incidence of type 2 diabetes. So when we think about sort of how we can treat these, I'm going to kind of jump back over here to type 1 diabetes, because that's, when I say fairly easy to treat, it's something that we just need to make sure that we provide the essential insulin that those individuals need. And so you'll see a lot of people nowadays, especially children, will have insulin pumps that not only help to monitor what their blood sugar levels are, but sort of in a AI feedback sort of way, provide the correct dosing of insulin. You know, prior to this was sort of a little bit of a guessing game in terms of, okay, you've consumed a certain amount of carbohydrate or sugar, you know, what is the amount of insulin that you need to take so that you can make sure that you properly allow that insulin to be taken up in your cell. You obviously don't want to provide too little insulin, because then you'll be left with high blood sugar, but you don't want to provide too much insulin, because then you'll put yourself in a hypoglycemic state. But with both of these situations, diet and exercise is really important, because diet and exercise really do help control your blood sugar levels, right? We've talked about that a lot. Ideally, you want to have, you know, as much as you can, sort of a fairly flat line of blood sugar levels. You know, clearly you eat, you're going to see an increase, you're going to secrete insulin, and then it's going to go down. What we don't want to see is a constant up and down cycling, because that is something that ultimately leads to insulin resistance. So for type 2 diabetes, right, diet and exercise is clearly very important. We need to make sure that we're monitoring the amount of carbohydrate that we're putting into our body, but exercise is really important. Early on, people tend to be treated with metformin. We're going to talk about metformin in a little bit. And again, later on, because they have this destruction or the inability of their own beta cells to produce insulin, they might need to start taking insulin themselves. And again, some other things here that are risk factors. I didn't mention here, sleep deprivation is an important risk factor for the development of type 2 diabetes, as is the use of statins and beta blockers. Low levels of testosterone are also known to be correlated with an increased risk for type 2 diabetes. And again, make sure that we note the difference between correlation versus causation. All right, so a lot on that slide. A lot of stuff I think is review, but again, a good review, kind of putting it together with this multi-organ system approach that we're kind of taking in this chapter. All right, so talk a little bit about metformin. Trade name is glucophage. This is the structure of it. So again, not a structure you need to know, but it's really interesting that it's very nitrogen rich, right? So this was actually found in 1922. It's a natural product found in French lilacs, and it was known for centuries, literally, as an important folk medicine component. So metformin is a first-line medication for type 2 diabetes. It's on the World Health Organization's list of essential medicines. And in 2017, it was actually the fourth most prescribed drug in the United States. So clearly a really important sort of drug. So we're going to spend a little bit of time talking about mechanism of action, particularly as it relates to concepts that we've already talked about. So here again are some things that it inhibits, okay? And it inhibits PKA, and it does so by decreasing levels of cyclic AMP. So again, when we've got cyclic AMP levels that have decreased, that means we're going to be inhibiting PKA. We talked about this very briefly in our electron transport and oxidative phosphorylation chapter. It inhibits complex 1. And remember, when we say it inhibits things, it doesn't mean it turns them off completely, but it suppresses the activity of complex 1, which means it decreases the activity of the electron transport chain. It also inhibits PepsiK and G6-PAse, and in doing so, it interferes and inhibits with gluconeogenesis. So let's see if we can kind of put all of these together and think about what that sort of means in terms of this overall process. So let's first just kind of think about the two things that I have down here in terms of stuff that is activated. And again, we're going to talk about AMPK in just a little bit here, but if we think about sort of things that are activated, and again, some of these, it's not well understood how it happens, but we actually can increase the uptake of glucose from the bloodstream, and metformin is known to do that by increasing the circulation of GLUT4 receptors to the surface. And then we're going to learn about this in a little bit, so we're going to activate AMPK, and AMPK kind of increases the concentration of AMP in the cytosol. And I'm not going to spend too much time talking about this piece right now because we really haven't learned about that enzyme yet. So let's just kind of like put all of these pieces together and see here are the main sort of things that happen. Metformin is used as a first-line medication for type 2 diabetics because it increases insulin sensitivity and it decreases glucose in the bloodstream. So let's see if we can understand how it decreases glucose in the bloodstream. Well, again, if we're decreasing gluconeogenesis and if we're inhibiting pKa, remember pKa puts us in a need state that's going to be doing things like breaking down glycogen. So if we make sure that we are inhibiting both bottom-up and top-down approaches to making glucose, we're not going to be offloading glucose from endogenous sources into the bloodstream. In addition, if we increase our GLUT4 transporters that are on the surface, that's going to help pull in glucose that's already in the bloodstream and get it into the cells. So again, these three sort of together help explain sort of the glucose drop in the bloodstream. And again, insulin sensitivity, again, that's not one of those things that's as well understood, as is the fact that it does seem to have an effect on the gut microbiome. And within the gut microbiome, it actually helps to decrease glucose absorption from your GI tract. So again, just finding a way to inhibit the amount of glucose that's going to get into your system. We're going to stop the production of it endogenously, and we're actually going to interfere with the absorption of it from an exogenous source. So let's talk a little bit about this enzyme, AMPK. When you think about, the K stands for kinase, right? It's an AMP-activated protein kinase. That's what those letters stand for. But I want you to think about K meaning conservation. And I know it's spelled wrong, but it's a kind of a funky way to help you remember that this is a conservation hormone. So we're going to activate this hormone when our metabolic demand outpaces our supply. So when we are in a low energy state, this hormone is going to kick in. This hormone is a hormone that's going to basically be upregulating any processes that are going to make ATP and downregulating any processes that are going to use ATP. So kind of like a situation where, let's say that you found out that you were sort of low on money, right? You're going to be finding ways that you might be taking on new jobs, doing whatever you can do to make more money, and you're going to stop spending money. So if ATP is really our biological money, we're going to be looking for all kinds of ways that we can make ATP, and we're going to be inhibiting any process that's not necessary that would use our ATP. So how is this whole process sensed and how does this work? Well, again, this ATP to ADP ratio, if we're in sort of a fed state, is going to be in about a 10-to-1 ratio. Okay? The ATP to AMP ratio in that same situation is going to be about 100-to-1. And so what we really have here is we have changes in AMP levels are going to be greatly magnified. We have a significant difference that we can see there. So again, AMPK responds here. So just like we had pKa, remember pKa, cyclic AMP is going to be its sort of signal. For AMPK, AMP is its signal, not cyclic AMP, just straight up AMP. So how do AMP levels get changed so much? Well, let's think about this. If we're in an energy need state, we're kind of dropping concentrations of ATP, right? We're going to have low ATP levels. So this ratio might be going down, but again, if it started out at 10 to 1, even if we're at 5 to 1, that's not that much of a change. But what happens is we have an enzyme called adenylate kinase that's going to kind of jump into play here. And it says, you know what? If we've got a need for ATP, how about this? I've got some ADP that's lying around. I have a good bit of it, but I'm going to actually do a substrate level phosphorylation where I'm going to take and sacrifice one of these ADPs and make it to AMP and regenerate one of them as an ATP. So you kind of start out with two things that are equal energy in their phosphorylation state. You elevate one of them to the complete sacrifice of the other. But why this is important too is it creates this important signal that allows us to then have this enzyme get kicked on. Okay, so again, what's going to happen here, we're going to look on the next slide just to kind of talk about a little bit of the structural details. But AMPK, in the presence of AMP, itself becomes phosphorylated and then can phosphorylate its downstream targets. So we're not going to go into too much detail here, but it is a protein that has 3 subunits to it. It has an alpha domain, which is the catalytic subdomain, and then it has 2 regulatory subunits, a beta and a gamma subunit. Again, these are the subunits that bind to AMP, ADP, or ATP. And basically, depending on what we have bound there, we actually change the conformation such that it changes the accessibility to threonine 172. So threonine 172 is that residue that would become phosphorylated with AMPK and become active. So again, if we've bound to AMP, we're basically going to be having a conformational change that exposes that threonine 172 here. It allows it to become phosphorylated and thus become active, and then it can go and phosphorylate its downstream targets. All right, so remember, a lot of these are phosphorylation cascades. So let's just introduce and ask the question, well, who actually does that important phosphorylation of that threonine residue? Who actually makes this conservation hormone be active? Well, we're going to learn about a couple of these here. We're not going to spend too much time on this liver kinase, but LKB1 is one of the kinases that can actually phosphorylate this threonine. But adiponectin is another one that can actually phosphorylate that threonine 172, and that in turn can make AMPK become active. I'm actually going to flip to the next slide to just kind of highlight this. We started talking about this, but I've got it kind of highlighted in a little bit neater way over here. All right, there's a lot to this slide, I know, but this is just kind of a way for us to see how important AMPK is. Okay, so again, we've got AMPK in an inhibited situation or an activated situation. Okay, so again, if AMPK is inhibited, okay, that means we are going to be inhibiting any pathways that would use ATP, right? If we're in conservation, we don't want to be spending our money. So we're going to inhibit any pathway that uses ATP. So let's look at some of these red arrows. Again, a great way for us to review things that we've talked about already and remind ourselves that some of these processes require ATP. So it's going to make sense that we're going to inhibit them. Okay, gluconeogenesis. We know that that's costly. So AMPK will inhibit gluconeogenesis, right? We've got the inhibition of sterol synthesis. That is actually fairly costly. Here's the enzymes that are responsible for that process. We've got that sterol recognition element binding protein, as well as HMG-CoA reductase. AMPK is involved in inhibiting both of those enzymes and thus the process of synthesizing sterols. Protein synthesis requires ATP. We didn't really talk too much about this, but if you've taken a class where you've sort of talked about sort of gene regulation or transcription and translation and so forth, when we talk about translation, which again is the process of synthesizing protein, remember you are going to have a whole bunch of tRNAs that are all primed with the cognate amino acid that they need to couple together and make the protein. So these tRNA synthetases, These are enzymes that are responsible for sort of loading up the corresponding amino acid on the tRNAs. This is a hugely costly process. So that's why protein synthesis requires so much energy, is because generating those building blocks for the ribosome to synthesize together, though that's really costly. So AMPK inhibits protein synthesis by inhibiting mTOR. Okay, we're going to talk about mTOR in a little bit, but mTOR stands for the mammalian target of rapamycin, and the C here stands for the fact that it's a complex. Okay. AMPK also is going to inhibit lipid synthesis, right? We learned about the rate controlling enzyme for fatty acid synthesis is ACC. If that enzyme is phosphorylated, it is not active. So we learned about that PKA can phosphorylate it, but AMPK can also phosphorylate it. This one's a little bit counterintuitive, but we actually are going to inhibit lipolysis. So it's a little bit weird because you're like, well, wait a minute, shouldn't we be like breaking down our fats so that we can go through beta oxidation and generate ATP? Yes, but the process of lipolysis, notice over here, we don't inhibit beta oxidation. What we're inhibiting is this cycle of converting fatty acids to triglycerides, to fatty acids to triglycerides. So what this really means is, you know what, we might not be as effectively tapping into the triglycerides that are in our adipose tissue, because kind of going through this process to kind of get them out of that storage and ultimately into a cell, that actually requires some energy. And so we are actually going to be inhibiting lipolysis here by AMPK. All right, so again, all of these processes are processes that would use ATP, so AMPK is going to be inhibiting them. And again, all of these we can consider to be using ATP because they are anabolic. They are building things, building glucose, building sterols, building protein, building lipid. All of those are costly, and so we are going to inhibit them. All right, so AMPK then is going to be activating any process that's going to be generating ATP. So in general, that's our catabolic processes. Okay, so we're going to be activating glycolysis. Notice here, if we activate glycolysis, it should make sense that we're going to be inhibiting gluconeogenesis. We're actually going to be activating glycogenolysis, right? We're going to activate phosphorylase so that we can break down glucose, and that glucose can then go into glycolysis. We're actually going to be helping with this process of glycolysis by facilitating the uptake of glucose from the bloodstream. So again, we're going to be recirculating GLUT4 receptors to the surface. We're going to be generating energy by beta oxidation, right? And again, if we're inhibiting ACC, that allows influx of fatty acids into the mitochondria so that we can undergo beta oxidation. So a couple of things that are a little bit new, maybe things we haven't talked about as much. We are actually going to also be upping our antioxidant defenses. If we're doing a lot of catabolism, we need to make sure that we've shored up our sort of antioxidant defenses so that we're not kind of putting ourselves in a situation where we're going to be subjecting ourselves to oxidizing stress. One of the other important things that AMPK does, we'll talk about this in a little bit, is called mitobiogenesis. So we basically are going to synthesize new mitochondria. I know that seems a little bit counterintuitive because it's like, wait a minute, I thought we weren't doing anything that synthesizes. But remember, we want to be lean and mean here. We want our biochemical processes to be working as effectively and efficiently as possible. So that might mean, kind of cleaning house on your mitochondria and making sure that the mitochondria that are there are working as best as possible. And so one of the processes that helps us clean house is this process called autophagy. Okay, so autophagy is a process that's going to allow us to basically get rid of any detritus, any things that are like not working properly, really just do some spring cleaning to make sure that we are lean and mean and kind of working at our maximum capacity. So some new hormones to talk about. And again, I've highlighted these in sort of red and green here because the ones that I've highlighted in red are going to be proteins that should inhibit hunger. So again, these should be fed state signals. Okay. In and of itself, leptin is often called the satiety hormone because high levels of leptin are supposed to make you feel full, not want to eat. It's actually secreted by your adipose cells, which is also why it's called a fat gene. So again, adipose cells, when again you're in a fed state, you've got lots of sort of energy around, your adipose cells are going to secrete leptin, and leptin acts on the hypothalamus and helps to inhibit hunger. Now, it's a little bit counterintuitive. They had some really interesting studies with mice that they actually found if you knocked out leptin, these mice would become obese. They would overeat and they would never feel satiated. They would never feel like they didn't want to eat. And so they constantly would eat. And so they would put on weight. So again, that's why this was called the fat gene. It was thought to be just kind of this amazing thing that they'd find with humans, and it was just going to be this cure to helping with obesity and all of that. Well, the problem is that it is secreted by adipose tissues, and what we actually see is that there's high level. It's like you would expect. If you've got a lot of adipose tissue, you're going to have a lot of leptin. Well, then the question becomes, why aren't these individuals then satiated? Why are they so hungry that they continue to overeat? And just like we see with insulin resistance, we see a hormonal resistance pathway here. We see that individuals who have obesity have this inability to detect satiety. And again, if you can't detect that you're satiated and you don't have an inhibition of hunger, you will overeat. And again, that leads to obesity. So again, elevated levels of individuals who are obese, about 30% elevation in leptin levels. So again, elevated leptin levels are sort of a proxy for obesity. And again, you're likely to then also have leptin resistance because you're not listening to that signal. Unfortunately, high levels of triglyceride also impair sort of the leptin receptor, so that's problematic. Elevated levels of fructose actually not only decrease the synthesis of leptin, right? Which again, if leptin is supposed to make you not feel hungry, if you have lots of fructose, you decrease synthesis of leptin, so it actually increases your hunger. But it also actually decreases leptin receptor synthesis. So again, I hope I've highlighted enough. We've got omega-3 fatty acids and we've got fructose. If you can avoid those, that's going to help on so many levels. All right, PYY over here. I'm not going to say too much about this. This actually stands for protein with 2 tyrosines in it. So that's why it's called PYY. It's a small hormone. It basically is secreted by your small intestine once you have food there. So the idea is it's a feedback loop, right? You've eaten food, it's gone through your stomach, made its way to the small intestine. So you're going to secrete this hormone, feed back to your hypothalamus, and make sure that you inhibit hunger so that you know you can stop eating. Okay? All right, 2 other hormones here that are highlighted in green because these are appetite stimulant hormones. So again, as much as leptin is called the satiety hormone, ghrelin is called the hunger hormone. This hormone is secreted by cells in your stomach. So again, just like we had, the hypothalamus is going to be this master regulator of what's going on in the environment. If your stomach cells are secreting ghrelin because your stomach is empty, it's saying, hey, you know what? I'm hungry. Let's send out a signal to make sure that we do things to satisfy that hunger. So one of the things that we talked about in class is the more stomach tissue that you actually have, the more ghrelin is secreted. And this is sort of counterintuitive. So if you perpetually overeat and your stomach stretches and you have more stomach tissue, you are going to find yourself more hungry because you just have more synthesis of ghrelin. So this is one of these things that they do find with people who have gastric bypass surgery, or if they have kind of a banding procedure or some way of decreasing the amount of stomach tissue that they have. They actually decrease the synthesis of ghrelin, and so that helps to suppress their appetite to kind of avoid this overeating. All right, adiponectin. Okay, again, this is an AMPK activator. Remember back here when we talked about who activates AMPK? There's adiponectin. So adiponectin is a kinase that's responsible for phosphorylating and thus activating AMPK. So again, this is something that's produced in adipose cells. So it's sort of the opposite of leptin. It is, interestingly enough, produced in adipose tissue, but it's inversely correlated with BMI. So for example, if you have a very healthy low BMI, you're actually going to have high levels of adiponectin. And so that's going to be really important. So that kind of puts you in this, not necessarily the conservation mode, but kind of puts you in a mode where you're not doing sort of anabolic synthesis processes. And so again, if you have low levels of adiponectin, that puts you at increased risk, not only for type 2 diabetes, but for that entirety of that syndrome that we call metabolic syndrome. And type 2 diabetes is, again, one of those components to it. So one of the things just to kind of highlight here, and this is just a figure that just kind of reminds us that it's all about energy balance. And even though it sounds really simple to just say, hey, it's just all about energy in, energy out, as long as you don't consume more calories than you need, you're going to be fine. But as we're seeing here, all of this is truly hormonally regulated, right? And so even though energy balance is really what it is about in terms of weight gain versus weight loss, right? It really has to do with whether you put yourself in an anabolic state, which will allow you to increase your food intake and then gain weight, or a catabolic state. Again, you're going to decrease your food intake and you're going to lose weight. And again, we've got these 4 hormones in addition to so many more, but really play kind of an important part in this entire process.